Hello there, and welcome to the Dreamy Ocean Channel. As you prepare to drift, allow your body to settle deeply into the quiet embrace of this moment. Feel the gentle release of any tension held within your shoulders, letting them soften and fall. Unclench your jaw, allowing your breath to flow in and out slow and steady, like the rhythmic hush of distant waves breaking upon a shore. Let your eyelids grow heavy as your thoughts begin to quiet, much like the ocean surface at dusk, reflecting the first stars. Tonight, we embark upon a serene journey into the hidden depths where the most exquisitely patterned forms of life whisper tales of both profound beauty and astonishing silent power. We will explore the delicate intricacies of shells, so captivating in their artistry that they have through time drawn curious human hands to their silent, perilous secrets, holding within them a potent, ancient stillness, a harpoon-like truth that lies beneath the polished surface. Allow your mind to float gently into these waters as the currents of discovery begin to carry us away into the deep. A deceptive beauty. The ocean's embrace, vast and ancient, holds within its shimmering depths a multitude of wonders each, a testament to the slow, deliberate artistry of time and evolution. Among these submerged treasures, there are certain forms that rise to the surface of human consciousness, not through their overt power, but through an almost ethereal beauty, drawing the eye with patterns so exquisite, so perfectly rendered, that they seem to be miniature universes held within a porcelain form. These are the shells of the cone snails, creatures of the warm, sun-drenched tropical waters, whose homes are often found nestled within the vibrant kaleidoscopic gardens of coral reefs, or partially buried beneath the fine, shifting sands of the intertidal zone, where the gentle ebb and flow of the tide whispers its eternal rhythm. To behold one of these shells is to witness a masterpiece of natural design, a silent sculpture crafted by a living organism over a lifetime of patient growth. Their forms are, as their name suggests, typically conical, tapering gracefully from a broader base to a delicate, often sharp apex, a shape that feels inherently balanced, aesthetically pleasing to the human hand and eye. The surfaces gleam, often possessing a smooth, almost polished quality reminiscent of fine ceramics or ancient ivory, reflecting the soft, diffused light of the underwater world in a way that suggests a deep inner luminescence. But it is in their coloration and patterning that these shells truly captivate, weaving intricate narratives across their curved exteriors. Each individual shell a unique scroll of abstract art. Imagine, if you will, a shell adorned with the deep, rich umber of ancient earth, swirling into the soft, sun-bleached cream of a forgotten shore. These tones often arranged in complex bands that encircle the cone, or perhaps in delicate, interconnected triangles, often described as tent patterns. These miniature apexes point towards the shell's graceful spire, creating an illusion of a tiny, forgotten village nestled upon a pristine porcelain landscape. The edges of these intricate markings might bleed softly into the surrounding background, a subtle blurring that speaks of countless aeons of evolutionary refinement, allowing the creature to dissolve visually into the dappled light and shadow of its coral reef home, a silent, almost invisible presence awaiting its moment. Other shells might display a mesmerizing checkerboard array of deep chocolate and pristine white, each square perfectly defined yet subtly softened by the ocean's gentle wear. Still others might be dappled with a constellation of fine, dark spots against a luminous golden background, evoking the image of a starry night sky, or painted with swirling eddies of deep violet and muted orange, as if a quiet sunset had been captured and solidified within their calcium carbonate walls. From the earliest days of human exploration and curiosity, these shells have been coveted. Ancient civilizations collected them for their beauty, using them as currency, as adornments, or as symbols of status and power, their intricate patterns whispering tales of distant, exotic lands. In more recent centuries, shell collecting evolved into a passionate pursuit, a scholarly endeavor, and a luxurious hobby. Wealthy collectors in the age of exploration would fill cabinets of curiosities with these oceanic jewels, each specimen a testament to the vast, unexplored wonders of the natural world. The act of finding a perfect, untouched cone snail shell on a remote beach became synonymous with discovering a hidden treasure, a tangible piece of the ocean's secret artistry, waiting to be admired, to be held, to be cherished. There is a profound, almost primal satisfaction in the act of discovery, in feeling the cool, smooth weight of such a shell in one's palm, tracing the elegant curves and delicate lines with a curious fingertip. The human eye is drawn to symmetry, to intricate detail, to vibrant or subtly blended colors, and the shells of the cone snails offer all of these in abundance. They appear to be inert, passive objects, relics washed ashore by the tireless tides, or resting peacefully on the sandy seabed, seemingly harmless and utterly captivating. This perceived innocence, this quiet beauty, is precisely where their profound deception lies. The very allure that draws a curious hand to reach down through the clear, sun-dappled water to gently retrieve one of these oceanic gems is a fundamental part of the creature's ancient, silent strategy. For beneath that polished, often vibrant exterior, within the seemingly tranquil confines of its elegant spire, resides a creature of astonishing predatory prowess. A hunter of the deep whose beauty masks a formidable and highly specialized weapon. The shell, so admired for its aesthetic grace, is not merely a home or a protective casing. It is an integral part of an elaborate, ancient camouflage, allowing its inhabitant to blend seamlessly with the complex textures and shifting lights of its environment. It becomes a silent sentinel, waiting with an almost infinite patience for its unsuspecting prey to drift within range, or for a curious hand to disturb its serene repose. Consider the scene: a tropical afternoon, the sun warming the shallow waters to a pleasant, inviting temperature. A beachcomber, perhaps, or a snorkeling enthusiast glides effortlessly over a patch of pristine white sand, dappled with the shadows of nearby coral. The water is so clear that every grain of sand, every miniature ripple, every tiny living detail of the seabed is visible with a startling clarity. And then there it is, a shell of exceptional beauty, perhaps larger than any seen before. Its patterns an intricate mosaic of earthy browns and creamy whites, lying partially obscured by a delicate frond of seaweed. The urge to reach out to gently lift this natural artwork from its watery bed is almost irresistible. The hand descends slowly, carefully, the fingers closing around the cool, smooth surface of the shell. There is a moment of pure wonder, a quiet satisfaction in possessing such a perfect specimen. But then, a subtle shift, a fleeting, almost imperceptible sensation like the brush of a fine, sharp needle, or the soft, sudden prick of a tiny thorn. It is not pain, not yet, but rather a momentary, unexpected sharpness, a pinprick that might be dismissed as a stray grain of sand or a tiny, harmless fragment of coral. There is no visible wound, no immediate rush of blood, no dramatic sign of injury. The shell remains smooth and inert in the hand, its pattern still mesmerizing, its form still elegant. The sensation might fade almost as quickly as it arose, leaving behind a faint, lingering tingling, a quiet numbness that spreads slowly, imperceptibly through the fingertips up the palm and into the wrist. This initial encounter, so often marked by an absence of overt alarm, is the very essence of the cone snail's deceptive beauty. The creature within, hidden by its magnificent shell, has delivered its silent warning, an almost imperceptible communication of its profound ancient power. The beauty of the shell, the very thing that draws the eye and beckons the touch, becomes the unwitting accomplice in a subtle yet potentially profound interaction. It is a testament to the quiet, often overlooked complexities of the natural world, where the most exquisite forms can conceal the most astonishing capabilities, reminding us that true understanding often lies far beneath the surface, beyond the initial glance, deeper than the first touch. The ocean, in its gentle, rhythmic breath, holds many such secrets waiting to be slowly, patiently unveiled, one intricate layer at a time. And as we continue our journey, allowing the currents of discovery to carry us gently forward, we will delve deeper into the astonishing mechanisms that lie beneath this beautiful, deceptive exterior, unveiling the cone snail's form. As the gentle currents of our journey carry us deeper past the shimmering outer beauty of the cone snail's shell, we begin to perceive the living architecture that resides within a soft, deliberate form that is the true artisan of its exquisite, calcified home. This creature, a gastropod mollusk, is not merely a passive inhabitant of its magnificent dwelling. It is an active, intricate being whose soft tissues are perfectly adapted to navigate sense and survive within the complex, vibrant tapestry of its underwater realm. To truly understand the cone snail, we must look beyond the gleaming patterns and delve into the quiet, purposeful movements of its hidden living form. From the aperture, the opening of the shell, a muscular foot emerges with a slow, almost imperceptible grace. This foot, often pale and translucent, sometimes adorned with subtle, mottled patterns that echo the shell's camouflage, is the primary means of locomotion for the cone snail. It is a remarkable organ capable of both adhering firmly to surfaces and gliding with an effortless fluidity. Imagine it as a broad, flattened sole, rippling with waves of muscle contractions that propel the snail forward across the sandy seabed or along the rough surfaces of coral. The movement is not swift, but rather a deliberate, almost meditative progression, a slow-motion ballet performed upon the ocean floor. Each ripple of the foot leaves behind a faint, almost invisible trail in the fine sediment, a temporary signature upon the soft canvas of the deep, a testament to its passing. As the snail extends its foot, its head region, small and often retracted, becomes more apparent. From this anterior part of its body, two delicate, slender tentacles extend like the antennae of a silent aquatic explorer. These tentacles are not merely for touch. They are sophisticated sensory organs constantly flickering, testing the surrounding water, tasting the subtle chemical gradients that drift through the currents. They are equipped with chemoreceptors, allowing the snail to detect the faintest molecular whispers of potential prey or the distant presence of a predator. Their movement is fluid, almost hypnotic, swaying gently with the water's ebb and flow, yet always purposeful, always gathering information, painting a silent chemical map of its immediate environment. At the base of these sensory probes, simple eyes are situated. These are not the complex image-forming eyes of a vertebrate. Rather, they are primarily light-sensing organs capable of distinguishing between light and shadow, detecting changes in ambient illumination, which can signal the approach of a larger creature or the shift from daylight to the soft twilight of dusk. Their gaze is not sharp, but rather a diffuse awareness, a quiet vigilance. Perhaps the most distinctive and crucial external appendage of the cone snail besides its shell is its siphon. This elongated, flexible tube, often extending significantly beyond the shell's aperture, is a marvel of biological engineering. It is a conduit, a living periscope that constantly samples the surrounding water, drawing it into the snail's mantle cavity, where the gills reside, facilitating the vital process of respiration. But its function extends far beyond mere breathing. The siphon is also densely packed with chemoreceptors, making it an exquisitely sensitive instrument for detecting prey from a distance. Imagine a delicate living hose continuously tasting the water, seeking the subtle chemical plumes released by fishworms or other mollusks creatures that might be hidden from view, but whose presence is betrayed by their molecular signatures. The siphon moves with a constant exploratory motion, sweeping arcs through the water like a silent underwater divining rod, its tip subtly flaring and contracting as it processes the rich information it gathers from the liquid world. This constant rhythmic sampling allows the cone snail, a creature of relatively slow movement, to pinpoint the location of its next meal with remarkable precision, even in the vast, opaque expanse of the ocean. Beneath the mantle, the soft tissue that lines the inside of the shell and is responsible for its growth and repair, lies the true heart of the cone snail's predatory genius, its venom apparatus. While the intricate details of the venom itself will unfold in later chapters, it is here within the very architecture of the snail's soft body that the mechanisms of its silent, potent weapon are housed. The most striking component of this system is the proboscis, a highly extensible muscular tube that can be rapidly averted from the mouth. When at rest, the proboscis is neatly retracted, an unassuming internal structure. But when activated, it can extend with surprising speed and length, sometimes reaching a distance comparable to the entire length of the snail's shell. This proboscis acts as a highly specialized delivery system for its unique weapon. Within the proboscis, a single modified Radular tooth is loaded ready for deployment. The Radular, a chitinous ribbon typically covered in rows of tiny teeth, used by most snails to scrape algae or drill into shells, has undergone a radical transformation in the cone snail lineage. In these creatures, the Radular has evolved into a series of individual, detachable, hollow harpoon-like teeth. Each tooth is a miniature, sophisticated projectile, crafted with a barb at its tip, designed to penetrate and anchor itself within the flesh of its prey. These teeth are produced in a specialized sac, the Radular sac, located within the snail's body, where they mature and are stored until needed. When a tooth is required, it is transferred from the sac into the proboscis, much like a dart being loaded into a blowgun or a spear into a launching mechanism. Connecting to this hollow Radular tooth and running through the proboscis is a venom duct, a channel that leads directly to the venom gland. This gland, a complex organ within the snail's body, is a sophisticated biochemical factory producing a potent cocktail of neurotoxins. The venom, once synthesized, is stored and concentrated within the gland, ready to be injected through the lumen of the hollow harpoon tooth. The entire system, the siphon detecting prey, the proboscis extending the tooth being loaded, and the venom being injected, represents an astonishing feat of evolutionary engineering. A perfectly synchronized sequence of events designed for maximum efficiency in a world where survival often hinges on a single, decisive strike. It is important to note that the cone snail's soft body is largely concealed within its shell, offering it protection from predators and the harsh elements of its marine environment. The mantle, a thin layer of tissue, not only secretes the shell material, but also encloses the mantle cavity, which houses the gills for respiration, and the osphradium, Another chemosensory organ that further refines the snail's ability to smell the water, confirming the presence and proximity of its target. When disturbed or threatened, the snail can rapidly retract its entire soft body back into the shell, sealing itself off from the outside world, with a remarkable swiftness, leaving only the beautiful silent shell to face any potential danger. While many snails possess an operculum, a hard protective trapdoor that seals the shell aperture, most cone snails have a greatly reduced operculum, or in some species, it is entirely absent. This suggests that their primary defense relies more on their potent venom and their ability to quickly withdraw and rely on the robust camouflaged shell itself for protection rather than a physical barrier. The cone snail, in its entirety, from the slow undulation of its foot to the delicate flick of its siphon, and the hidden power of its venom apparatus, is a testament to the quiet ingenuity of life in the ocean. It is a creature designed for stealth and precision, a silent architect of its own survival, crafting not only a beautiful, deceptive shell, but also an intricate, potent, biological weapon. The seeming passivity of its shell belies the vibrant, active, and profoundly dangerous life contained within a life exquisitely attuned to the subtle rhythms and chemical whispers of the deep. As we allow our thoughts to gently drift further into this fascinating world, we begin to appreciate the layers of complexity that lie beneath the surface, preparing to explore the very essence of its remarkable, silent power. Patterns of deception and grace. The shell, that exquisite outer dwelling, is far more than a mere protective casing. It is a silent, enduring testament to the vast, slow processes of life. Beneath the waves, a promise whispered in calcium carbonate and pigment. Each curve, each subtle undulation, each meticulously rendered pattern upon its surface tells a story of survival, of adaptation, and of a profound, ancient deception. These shells are not simply found, they are grown layer by painstaking layer by the soft, living mantle tissue of the snail, within a process that unfolds with an almost geological patience, adding infinitesimal increments of material over the creature's entire lifespan. Imagine the mantle, a delicate, fleshy membrane that lines the interior of the shell, acting as a living sculptor. From its specialized glands, it continuously secretes calcium carbonate, the fundamental building block of the shell, often in the crystalline form of aragonite, interspersed with a matrix of organic proteins. This slow, steady deposition of material occurs along the growing edge of the shell's aperture, gradually expanding its circumference and adding new coils to its elegant spiral. With each passing day, each cycle of nutrient intake and metabolic activity, the shell extends outward, subtly increasing in size, its form, spiraling gracefully around a central axis known as the columella. The visible lines of growth, often subtle ridges or faint concentric rings, are like the annual rings within an ancient tree, silently recording the passage of time, the periods of abundant growth, and the quiet pauses in its steady expansion. Upon this foundation of mineral and protein, the true artistry of the cone snail unfurls. The outermost layer of the shell, particularly in live specimens or those freshly washed ashore, is often covered by a thin organic skin called the periostracum. This layer, typically a duller brown or green, can offer an initial layer of camouflage, sometimes bearing fine hair-like projections or subtle textures that help the shell blend into its environment, perhaps mimicking the fine filaments of algae or the grainy texture of sand. Over time and through the gentle, abrasive action of waves and currents, this delicate periostracum often wears away, revealing the breathtaking polished surface and vibrant patterns beneath the very patterns that beckon with such compelling allure. The shape itself, typically a smooth inverted cone, is a marvel of hydrodynamic efficiency and structural integrity. Its broad base provides a stable platform for the snail's muscular foot, allowing it to navigate the varied terrain of the seabed, while the gentle taper of its spire minimizes resistance to the surrounding water currents. This conical form is remarkably strong, capable of withstanding the crushing pressures of the deep or the relentless battering of waves in shallower zones, protecting the soft, vulnerable body within from the jaws of predators or the accidental impact of shifting rocks. The aperture, the wide opening from which the snail emerges, is often elongated and narrow, a strategic design feature that provides a secure internal space for retraction, while the siphonal canal, a distinct notch or channel at the shell's anterior end, provides a specialized passage for the crucial siphon, allowing it to extend and sample the water without exposing the entire body. But it is in the patterns, the vibrant calligraphy etched upon the shell's surface, that the cone snail's deceptive grace finds its most profound expression. These patterns are not randomly applied. They are meticulously laid down by specialized cells within the mantle, which deposit pigments during the shell's growth. The specific combination of pigments, their concentration, and the precise timing of their release create an astonishing diversity of designs, each unique to the species, and often subtly varying even within individuals of the same species. Consider, for instance, the archetypal tent patterns, so named for their resemblance to rows of miniature interconnected triangles, often in shades of deep brown, rich ochre, or velvety black set against a background of creamy white, pale yellow, or soft grey. These patterns are not merely decorative, they are a masterclass in visual dissolution. When viewed against the dappled sunlight filtering through shallow water, or nestled among the broken fragments of coral and shell rubble on the seabed, these sharp, angular markings effectively break up the snail's outline, making it incredibly difficult for a discerning eye to perceive the continuous form of the shell. The contrasting colors and sharp geometries mimic the fragmented shadows and varied textures of the reef floor, allowing the snail to become, for all intents and purposes, invisible to both its prey and its predators. It becomes a silent, waiting presence, a master of disguise. Other species display broad, sweeping bands of color that encircle the cone like ancient painted pottery. These bands might range from deep, resonant purples to fiery oranges, from muted greens to stark, contrasting blacks and whites. These patterns, too, serve a purpose beyond mere aesthetics. In some cases, these bold bands might aid in disruptive coloration, creating an illusion of movement or breaking up the shell's solid form when viewed from a distance. In others, particularly those with less intricate designs, the specific coloration might allow the snail to blend into particular substrates, such as the dark volcanic sands of certain regions, or the vibrant hues of specific algal beds. Imagine a shell of deep oceanic blue, almost indistinguishable from the shadows cast by an overhanging coral head, or a specimen patterned with soft, earthy greens and browns, melting seamlessly into a bed of seagrass. Then there are the shells adorned with intricate reticulated patterns, a delicate network of fine lines that crisscross the surface, forming a complex, almost lace-like grid. These patterns might be in contrasting colors, creating a subtle three-dimensional effect, or in closely related shades, giving the shell a soft, almost blurred appearance. Other shells are dappled with scattered spots or blotches, like a constellation of tiny stars upon a dark background, or the random scattering of pebbles on a pristine beach. Each dot, each blotch, each intricate line is placed with a precision that speaks of countless generations of evolutionary fine-tuning, ensuring that the shell's visual signature is perfectly adapted to its specific ecological niche. While camouflage is widely accepted as the primary function of these diverse patterns, scientists also ponder other potential roles. It is theorized that certain bold or contrasting patterns might, in some instances, serve as a mild form of aposematism, a warning signal to potential predators subtly indicating the snail's venomous nature without resorting to the overt bright colors often seen in terrestrial venomous creatures. However, this is a more nuanced hypothesis, as the primary strategy of the cone snail appears to be stealth rather than overt warning. Furthermore, the intricate patterns and unique coloration might also play a role in species recognition, allowing individual cone snails to identify and differentiate members of their own species for reproductive purposes, a subtle visual cue in the vast, often monochromatic world of the seabed. The surface texture of these shells also contributes to their deceptive grace. Many species possess an incredibly smooth, almost porcelain-like finish, which not only gives them their coveted luster, but also helps to prevent the attachment of fouling organisms, such as algae or barnacles, which could compromise their camouflage or impede their movement. Other species might exhibit subtle sculptural elements such as fine spiral grooves, delicate axial ribs, or small raised nodules, particularly around the spire or shoulder of the shell. These textural variations add another layer of complexity to their camouflage, allowing the shell to mimic the rough, irregular surfaces of coral fragments or the gritty texture of coarse sand, further enhancing its ability to disappear within its environment. Ultimately, the shell of the cone snail is a silent promise, an exquisite paradox. It promises beauty, a moment of aesthetic wonder for the human observer, drawing the eye with its flawless form and intricate artistry. Yet this very beauty is a profound deception, a perfectly evolved camouflage that conceals a quiet, potent danger. The shell, so seemingly inert and passive, is an active participant in the snail's survival strategy, a masterpiece of natural engineering that allows its venomous inhabitant to remain hidden, to hunt with precision, and to protect itself from the myriad challenges of the deep. As we allow our minds to continue their gentle drift, we carry with us the image of these silent patterned jewels, understanding now that their grace is not merely aesthetic but a fundamental component of their powerful ancient truth. Anatomy of the Radular tooth. Beyond the elegant deception of the shell nestled deep within the intricate anatomy of the cone, snail lies the true heart of its formidable power, a weapon of unparalleled specialization, a silent, microscopic spear. This is the Radular tooth, not merely a tooth in the conventional sense, but a finely engineered projectile, a hollow harpoon crafted with astonishing precision, designed for a single decisive purpose: the swift and silent delivery of potent venom. To truly appreciate the genius of this creature, we must trace the journey of this remarkable structure from its nascent formation to its moment of lethal deployment. In most gastropods, the radula is a ribbon-like organ adorned with rows of chitinous teeth, functioning much like a flexible rasp or file used to scrape algae from rocks or bore into the shells of other mollusks. But in the lineage of the cone, snail's evolution with its infinite patience and relentless refinement has transformed this ancient tool into something entirely new, something radically adapted for active predation. Here, the radular ribbon has been reduced, its numerous teeth distilled into a series of individual detachable units, each a miniature masterpiece of biological weaponry. Imagine a specialized internal factory, a delicate assembly line within the snail's soft body, known as the Radular sac. Within this elongated internal chamber, often resembling a slender, coiled tube, the radular teeth are continuously produced one after another in various stages of development. At one end of this sac, the earliest immature teeth begin to form, gradually maturing as they slowly progress along its length, much like a meticulous craftsman perfecting each successive piece. These teeth are composed primarily of chitin, a durable, flexible biopolymer similar to the material found in insect exoskeletons. However, to enhance their piercing capability and structural integrity, scientists believe that some cone snail species incorporate trace amounts of metallic elements, such as zinc or iron, into the very tip or shaft of the tooth, subtly increasing its hardness and rigidity, allowing it to penetrate tough hides or scales with minimal deformation. As each tooth matures, its form solidifies into the characteristic harpoon shape. Visualize a slender, hollow shaft, often tapering gently from a slightly broader base to an incredibly fine needle-sharp point. This point is not merely sharp, it is honed to a microscopic precision, capable of cleaving through the resilient scales of a fish, the muscular flesh of a worm, or the delicate tissues of another mollusk, with astonishing ease, minimizing resistance and ensuring rapid penetration. Just behind this acute tip, a barb or a series of recurved serrations are often sculpted into the chitin. This barb is a critical feature serving a dual purpose. Firstly, it ensures that once the tooth has pierced its target, it anchors itself firmly within the flesh, preventing easy dislodgement. Secondly, it provides a secure channel for the venom, ensuring that the potent cocktail is delivered deeply and effectively into the prey's circulatory system or nervous tissue. The most crucial anatomical feature of this tooth is its hollowness. A microscopic lumen, a narrow internal channel, runs the entire length of this chitinous shaft, from its base to its finely sharpened tip. This internal conduit is the pathway for the venom, the silent liquid messenger of paralysis. The base of the tooth is often slightly flared or possesses a specialized structure that allows it to be securely connected to the proboscis, the extensible muscular tube that acts as the delivery mechanism. This connection is not permanent. It is designed for a precise temporary coupling, allowing the tooth to be swiftly deployed and then crucially detached after its mission is complete. Within the radular sac, a supply of these mature, ready-to-use teeth is meticulously stored, arranged in a linear series like a quiver, holding a succession of finely crafted arrows. This continuous production and storage ensure that the cone snail always has a fresh, potent weapon at its disposal, ready for its next hunt. When the snail detects a suitable prey item through the sensitive chemoreceptors of its siphon, a remarkable sequence of events is initiated. A single mature radular tooth is selected from the radular sac and transferred with precise muscular contractions into the proboscis, the extensible muscular tube that will act as the gun barrel for this living spear. This loading process is swift and efficient, ensuring that the weapon is prepared for immediate deployment. The proboscis itself is an extraordinary organ capable of rapid extension and retraction. When at rest, it is compactly folded within the snail's body. But in the presence of prey, it can be everted and elongated with surprising speed, sometimes reaching a length greater than the snail's own shell. As the proboscis extends, it guides the loaded radular tooth towards the unsuspecting target. The moment of strike is often incredibly fast, a sudden, precise thrust. The proboscis, with the harpoon-like tooth at its tip, is jabbed into the prey. At the instant of penetration, the venom gland, a specialized organ located within the snail's body and connected to the hollow tooth via a venom duct, contracts forcefully. This contraction injects the stored venom through the lumen of the hollow tooth directly into the tissues of the prey. The entire process from detection to injection is a masterclass in predatory efficiency, a silent, almost instantaneous act of chemical warfare. Once the venom has been delivered, the Radular tooth detaches from the proboscis. In many species, the tooth remains embedded within the prey, acting as a continuous delivery system for the venom or simply as a marker of the successful strike. The proboscis then retracts, and the snail, often moving slowly, begins to track its now paralyzed or rapidly weakening victim, using its sensitive chemoreceptors to follow the subtle chemical trails left by the venom's effects. A new tooth is then loaded into the proboscis from the ready supply in the Radular sac, ensuring that the hunter is always prepared for its next opportunity. The morphology of these Radular teeth is incredibly diverse, reflecting the vast array of prey items that different cone snail species target. For example, fish-hunting cone snails known as piscivores typically possess larger, more robust teeth with prominent barbs designed to penetrate the tough scales and strong muscles of fish and hold them securely. Worm-hunting species or vermivores often have more slender, finely serrated teeth optimized for piercing the softer, segmented bodies of marine worms. Mollusk-hunting species or molluscivores may have teeth with broader bases or more complex shapes adapted to either drill into or hook into the shells or soft tissues of other snails. This remarkable diversity in tooth design is a clear indicator of the highly specialized nature of each cone. Snail species, each having evolved a unique, silent spear, perfectly suited to its particular hunting strategy and preferred diet. The Radular tooth, therefore, is not a simple biological tool. It is a complex, elegant weapon, a testament to millions of years of evolutionary refinement. Its production, its storage, its precise loading, and its swift deployment are all orchestrated with an astonishing biological precision, making it the central component of the cone snail's predatory success. It is the very essence of its silent, potent power, a miniature harpoon that through its hollow core delivers a profound and often fatal chemical message, transforming the beautiful, seemingly passive shell into the home of one of the ocean's most sophisticated and efficient hunters. As we continue to delve into the depths of this creature's astonishing biology, we will next explore the very substance that flows through this silent spear. The venom itself, a liquid labyrinth of peptides and paralysis. A miniature pharmaceutical factory. Having explored the precise mechanics of the Radular tooth, that silent chitinous spear, our journey now turns inward into the very heart of the cone snail's chemical arsenal, the venom gland. This remarkable organ, often slender and elongated, coiling gently within the soft tissues of the snail's body, is a miniature pharmaceutical factory, a biological marvel that operates with astonishing efficiency and complexity, continuously synthesizing a potent cocktail of neurotoxins. It is here in this hidden, unassuming chamber that the liquid labyrinth of paralysis is meticulously crafted, drop by potent drop. Imagine a specialized internal tube, sometimes several times the length of the snail itself, folded and tucked away, its inner surface lined with a rich tapestry of highly specialized secretory cells. These cells are the tireless workers of this biochemical factory, each one a microscopic marvel dedicated to the intricate task of building complex molecules from simpler components. The gland is not a single homogeneous entity. Scientists believe it often comprises distinct regions, perhaps a proximal end where the initial synthesis begins, and a distal end where the venom matures, and is stored, each segment playing a crucial role in the overall production line. At the very foundation of this factory's operation lies the genetic blueprint encoded within the snail's DNA. Within the nucleus of each secretory cell, specific genes, those dedicated to venom production, are quietly activated. This activation initiates the first step transcription, where the genetic information from DNA is carefully copied into molecules of messenger, RNA, or mRNA. These mRNA strands, carrying the precise instructions for building venom components, then drift out of the nucleus and into the cytoplasm of the cell, where the true work of construction begins. In the cytoplasm, these mRNA blueprints encounter the ribosomes, the cell's protein-building machinery. These tiny, intricate molecular complexes act as assembly stations, reading the instructions on the mRNA and precisely linking together amino acids, the fundamental building blocks of proteins and peptides. As these amino acids are strung together, they form long, nascent polypeptide chains, the raw, unfinished forms of the future conotoxins. This process, known as translation, is a testament to the cell's remarkable ability to transform genetic code into tangible, molecular structures, a silent, continuous hum of creation. But the creation of these venom peptides is far from complete. The freshly synthesized polypeptide chains are still linear, unorganized strands devoid of their potent biological activity. They must undergo a series of crucial transformations known as post-translational modifications, which are absolutely essential for their function. These modifications occur within other specialized compartments of the cell, particularly the endoplasmic reticulum and the Golgi apparatus, which act as the cell's intricate processing and packaging centers. Within the endoplasmic reticulum, these nascent peptide chains begin to fold into precise three-dimensional structures. This folding is not random. It is guided by the specific sequence of amino acids and assisted by molecular chaperones, which are like patient, unseen hands, gently coaxing the chains into their correct, biologically active conformations. A critical step in this folding process for many conotoxins is the formation of disulfide bonds. Imagine tiny molecular bridges forming between specific sulfur-containing amino acids within the peptide chain, locking the molecule into a stable, rigid structure. These disulfide bonds are incredibly important. They are the molecular anchors that give many conotoxins their remarkable stability and their precise target specificity, ensuring that they can withstand the rigors of injection and interact with their intended molecular targets in the prey with exquisite accuracy. Following the initial folding and disulfide bond formation, the peptides often move into the Golgi apparatus, a series of flattened membrane-bound sacs. Here they undergo further processing, which can include proteolytic cleavage. This is like a precise molecular scissors, cutting larger precursor proteins into smaller active peptide fragments. This step is crucial for generating the final potent conotoxins from longer inactive protein chains, much like refining a raw material into a finished, powerful product. In addition to cleavage, other modifications might occur, such as glycosylation, the attachment of sugar molecules, or hydroxylation, the addition of hydroxyl groups, further diversifying the chemical landscape of the venom components. Once these peptides have been fully synthesized, folded, modified, and refined, they are not immediately released. Instead, they are meticulously packaged into tiny membrane-bound vesicles, like miniature storage containers within the secretory cells. These vesicles then migrate towards the lumen, the central channel of the venom gland, where they fuse with the cell membrane and release their precious cargo. The venom now, a complex concentrated solution of active conotoxins, begins to accumulate within the gland's lumen, awaiting the moment of its deployment. The sheer diversity of conotoxins produced by a single cone snail species is staggering. A single gland can produce hundreds, sometimes thousands of distinct peptide toxins. This is not a simple single-component venom. It is a highly sophisticated cocktail, a liquid labyrinth of different peptides, each designed to target specific molecular pathways within the prey's nervous system. Some conotoxins might block ion channels, preventing nerve impulses from firing. Others might interfere with neurotransmitter release, disrupting communication between nerve cells. Still others might act on muscle receptors, inducing rapid paralysis. This multi-component approach ensures that the venom is incredibly effective, often causing rapid immobilization or death, overwhelming the prey's physiological systems through multiple pathways simultaneously. Scientists believe that the cone snail's venom gland operates with a remarkable degree of dynamic control. The specific composition of the venom cocktail can vary not only between different species, but also within a single individual, potentially influenced by factors such as the snail's age, its diet, or even the environmental conditions. This adaptability allows the snail to fine-tune its chemical weapon, optimizing its effectiveness against the specific types of prey it encounters most frequently, or perhaps even adjusting its venom profile in response to different predatory challenges. It is a constantly evolving living pharmaceutical factory, subtly adjusting its output to meet the demands of its silent predatory life. The accumulated venom, a potent concentrated liquid, is then stored within the distal regions of the gland, ready for immediate use. A muscular bulb, often associated with the venom duct, acts like a powerful pump. When the snail strikes this muscular bulb, contracts forcefully, generating the pressure required to propel the stored venom through the narrow venom duct, down the length of the proboscis, and out through the hollow lumen of the radular tooth, directly into the tissues of the unsuspecting prey. This entire system, from the genetic instructions to the final forceful injection, represents an astonishing synergy of biological design, a testament to the power of evolution to craft such intricate and effective biological weapons. Thus, the venom gland is not merely a reservoir, it is a dynamic, living laboratory, a silent subterranean factory churning out an array of highly specialized chemical tools. It is the engine behind the cone snail's reputation as a beautiful yet deadly hunter, transforming the very essence of its life into a potent, paralyzing force. As we continue our gentle drift, carrying with us the understanding of this hidden factory's intricate workings, we are now prepared to explore the astonishing effects of its liquid creations, the conotoxins themselves, and the profound impact they have upon the nervous systems of their prey. The liquid labyrinth of peptides and paralysis. From the hidden depths of that miniature pharmaceutical factory, the venom gland emerges a truly astonishing creation: the conotoxins. These are not simple singular poisons, but rather an intricate shimmering array of small proteins and peptides, each a molecular key crafted with exquisite precision, designed to unlock and disrupt specific vital functions within the nervous systems of their prey. To understand the cone snail's silent power is to understand this liquid labyrinth, this complex mixture of biochemical whispers that collectively orchestrate a profound and often rapid paralysis. Imagine, if you will, a vast, intricate network within any living creature, the nervous system. It is a ceaseless flow of electrical signals and chemical messages, a grand, silent conversation between billions of cells, governing every thought, every movement, every heartbeat. At the heart of this communication are specialized proteins embedded within the membranes of nerve cells, and muscle cells acting as gatekeepers and receivers. These are the ion channels, which control the flow of electrically charged atoms or ions across cell membranes, thereby generating electrical impulses. And these are the receptors, which are like tiny molecular antennae, designed to specifically bind to chemical messengers called neurotransmitters, thereby relaying information from one cell to another. The conotoxins, in their astonishing diversity, are masters at interfering with these fundamental processes, each peptide targeting a specific gate or antenna with uncanny accuracy. Consider the alpha conotoxins, a group of peptides renowned for their ability to specifically target nicotinic acetylcholine receptors. These receptors are absolutely crucial for the transmission of signals from nerve cells to muscle cells, particularly at the neuromuscular junction, the specialized synapse, where a motor neuron communicates with a muscle fiber, telling it to contract. When an alpha conotoxin enters the bloodstream of a fish or a worm, it silently drifts towards these receptors. Picture it as a perfectly shaped molecular imposter mimicking the natural neurotransmitter acetylcholine. But instead of activating the receptor, it binds to it tightly, like a key stuck in a lock, preventing the real acetylcholine from attaching. This blockage means the nerve's command to the muscle cannot be heard. The muscle, deprived of its signal, remains flaccid, unable to contract. The fish, once darting through the water, becomes suddenly still, its fins losing their power, its body slumping into an unmoving, silent form. The worm, once wriggling, becomes limp, its segments unresponsive to the urgent commands from its own nervous system. Then there are the mu conotoxins, another potent class, whose primary targets are the voltage-gated sodium channels. These channels are the very conduits through which nerve impulses, those rapid electrical signals, propagate along nerve fibers. When a nerve cell fires, sodium channels open, allowing a swift influx of positively charged sodium ions, creating an electrical current that races down the nerve, carrying its message. Mu conotoxins act as highly specific blockers, binding to these channels and preventing them from opening. It is like placing a tiny invisible plug into each of these vital conduits. The nerve impulse, unable to flow, simply ceases. The message cannot be sent. The sensation for the prey might begin as a strange numbness, a creeping loss of feeling, gradually spreading into an inability to initiate any movement. The muscles, though not directly targeted at the receptor level, become paralyzed because they never receive the electrical command to contract from the silenced nerves. Other conotoxins, such as the omega conotoxins, focus their attention on voltage-gated calcium channels. These channels are critical for the release of neurotransmitters at the nerve terminals, the very points where one nerve cell communicates with the next. When an electrical impulse arrives at a nerve terminal, calcium channels open, allowing calcium ions to rush in, triggering the release of neurotransmitters into the synaptic cleft, the tiny gap between nerve cells. Omega conotoxins, by binding to and blocking these calcium channels, effectively shut down this crucial release mechanism. The nerve impulse might still arrive at the terminal, but the chemical messenger that bridges the gap to the next cell or to the muscle can no longer be discharged. This leads to a profound disruption of synaptic transmission, causing a widespread weakening and eventual paralysis of muscles as communication across the nervous system falters and then ceases entirely. And the labyrinth extends further still. There are delta conotoxins, which do not block channels, but rather keep sodium channels open for an extended duration, causing an over-excitation of nerves that quickly leads to paralysis as the nerve cells become depolarized and unable to fire again effectively. There are kappa conotoxins targeting potassium channels, which are vital for repolarizing nerve cells after they fire, helping them reset for the next impulse. By interfering with these, the delicate rhythm of nerve firing is thrown into disarray, leading to further dysfunction and paralysis. Still others, the iota conotoxins, have been found to affect calcium release channels in muscle cells, directly disrupting the internal machinery that allows muscles to contract. The astonishing feature of the cone snail's venom is not just the sheer number of distinct conotoxin peptides. A single species can produce hundreds, sometimes thousands of different ones, but their incredible specificity. Each peptide is often designed to interact with a particular subtype of ion channel or receptor found in specific tissues or specific organisms. This is why different cone snail species targeting different prey have evolved distinct venom cocktails. For the piscivorous or fish-hunting cone snails, the venom must be incredibly fast-acting. Fish are swift, agile creatures, and a slow-acting venom would allow them to escape, perhaps to die later, but far from the waiting snail. Thus, the venom of fish-hunting cone snails, such as Conus geographus, contains a potent mix of toxins designed to induce immediate, profound paralysis. The onset of effects can be almost instantaneous, causing the fish to become rigid or flaccid. Within seconds, its frantic struggles ceasing abruptly, allowing the relatively slow-moving snail to engulf its helpless meal. These venoms often contain a high concentration of alpha-conotoxins and mu-conotoxins, ensuring rapid blockade of both muscle receptors and nerve conduction, leading to a swift, overwhelming incapacitation. Conversely, vermivorous or worm-hunting cone snails often employ venoms with a slightly different profile. Marine worms, while not as swift as fish, can burrow deeply into the sand or mud, making them difficult to retrieve if they are not quickly immobilized. The conotoxins in these venoms might be designed to induce a more sustained, perhaps even spastic paralysis, preventing the worm from retreating into its burrow, or causing it to emerge from its hiding place. The mix might include peptides that cause prolonged muscle contraction or sensory disruption, making escape impossible. Molluscivorous cone snails, those that prey on other snails or bivalves, face yet another challenge, overcoming the hard shells or strong adductor muscles of their prey. Their venoms contain conotoxins tailored to disrupt the specific physiological processes of other mollusks, perhaps affecting their ability to retract into their shells or to firmly clamp their bivalve shells shut. The precise molecular targets in other mollusks are still an area of active research, revealing the depth of this biochemical arms race. The true genius of the cone snail's venom lies in the synergistic action of these diverse peptides. It is not just the presence of individual toxins, but how they work together. Like a finely tuned orchestra of chemical disruption, each playing its part to achieve a devastating effect. One conotoxin might block sodium channels, silencing nerve impulses, while another simultaneously blocks acetylcholine receptors, directly paralyzing muscles, and yet another might disrupt calcium channels, preventing neurotransmitter release. This multi-pronged attack overwhelms the prey's nervous system through multiple pathways simultaneously, ensuring a rapid, robust, and irreversible incapacitation. The effect is far greater than the sum of its individual parts, a testament to millions of years of evolutionary refinement. This liquid labyrinth, once injected into the unsuspecting prey, begins its silent work. There is no visible wound, no dramatic sign of injury, only the gradual, insidious spread of numbness, followed by weakness, and then the profound stillness of paralysis. The prey, once vibrant and full of life, becomes a quiescent form. Its movements stilled its senses, dulled its struggle. ended not by brute force but by the whisper of molecular keys turning in vital locks. It is a powerful reminder that in the quiet depths of the ocean, the most exquisite beauty can conceal the most sophisticated and potent forms of silent chemical power. As we allow our thoughts to gently drift further, we will consider the moments when this silent spear and its potent liquid encounter an altogether different and often tragic fate: the unwary human. Hunting strategies in the ocean depths. Having understood the liquid labyrinth of conotoxins, those exquisitely crafted molecular keys, our journey now turns to witness their profound purpose: the silent, deliberate act of hunting a complex ballet of stealth, precision, and chemical warfare that unfolds within the quiet depths of the ocean. The cone snail, despite its seemingly placid exterior, is a master predator, each species having evolved a unique suite of strategies finely tuned to the specific vulnerabilities of its chosen prey. These are not creatures of swift pursuit, but rather architects of the perfect ambush, relying on an ancient patience and an astonishingly potent weapon to secure their silent, paralytic embrace. Consider first the strategy of the lie-in-wait predator, a common approach among many cone snail species, particularly those that target swift-moving fish. Imagine a stretch of pristine white sand, perhaps dappled with the shifting shadows of coral branches above, or partially obscured by a delicate frond of seagrass. Here, a cone snail might slowly, meticulously bury itself, sinking gently into the fine sediment, until only the very tip of its elongated siphon protrudes like a miniature living periscope. Its shell, with its intricate patterns of deception, is perfectly camouflaged against the textured seabed, rendering the creature almost invisible to the casual glance of a passing fish. Time becomes an irrelevant concept for this patient hunter. It can remain motionless for hours, even days, conserving its energy, allowing the subtle currents to carry the chemical whispers of the ocean to its highly sensitive sensory organ. The siphon, that remarkable constantly flickering tube, continuously samples the surrounding water, drawing in microscopic molecules and analyzing their chemical signatures. It is tasting the water, searching for the faint telltale plumes of a fish, perhaps the subtle release of amino acids from its gills or the delicate scent of its scales. A fish unaware of the hidden danger might swim languidly by its fins, undulating with a rhythmic grace, its body reflecting the filtered sunlight. As it draws closer, the chemical signals intensify, stimulating the snail's internal alarm. A quiet tension builds within the buried hunter, a subtle shift in its internal chemistry, preparing for the decisive moment. When the unsuspecting fish is within striking distance, a precise, calculated range, determined by the snail's species. Specific proboscis length, the ambush is sprung. The proboscis, that highly extensible muscular tube which has been held, coiled and retracted, now extends with astonishing speed, a sudden, almost explosive unfolding. From its tip, the single hollow harpoon-like Radular tooth is rapidly thrust forward, piercing the fish's flesh with a needle-sharp precision. At the exact moment of penetration, the venom gland contracts, forcefully injecting its potent cocktail of conotoxins through the hollow lumen of the tooth, directly into the fish's body. The entire strike can occur in a fraction of a second, a blur of motion too swift for the human eye to fully register, leaving the fish no time to react, no opportunity for escape. The effect of the venom is often immediate and profound. For fish-hunting cone snails, the conotoxins are designed for rapid, overwhelming paralysis. The fish moments ago swimming freely might suddenly become rigid, its muscles locking in a spastic contraction, or it might go completely limp, its body slumping its fins, ceasing their rhythmic movement. This rapid incapacitation is crucial. A struggling fish could easily escape the snail's grip or injure the proboscis. The venom ensures that the prey is rendered utterly helpless, a silent, unresisting meal allowing the slow-moving snail ample time to engulf its catch. Once the fish is paralyzed, the radular tooth detaches, and the proboscis, often acting like a wide elastic net, slowly engulfs the fish whole, drawing it into the snail's distended mouth. A process that can take many minutes, a deliberate, unhurried consumption. One of the most remarkable and specialized hunting strategies is employed by Conus geographus, often referred to as the cigarette snail, due to the grim old adage that a human victim might only have enough time to smoke one cigarette before succumbing to its venom. This species is a prime example of the net hunting strategy. Instead of simply thrusting its proboscis, Conus geographus extends its proboscis into a wide, gaping, funnel-like structure, a living, muscular net. It then slowly approaches a school of small fish or positions itself near a resting fish, often at night, when fish are less active. The proboscis, now a wide, inviting maw, might even release a chemical attractant, a subtle lure to draw the fish closer. When a fish swims into this expanded proboscis, the snail rapidly contracts the muscular net, trapping the fish within. Only then is the venomous radular tooth deployed, piercing the struggling fish multiple times, delivering a swift, overwhelming dose of toxins specifically designed to induce almost instantaneous paralysis. This strategy allows Conus geographus to capture multiple fish in rapid succession, or to secure a larger, more active prey item that might otherwise evade a direct harpoon strike. Worm-hunting cone snails or vermivores exhibit a slightly different approach. While many also employ ambush tactics, some species are more active foragers. They might slowly glide across the sandy or muddy seabed, their siphons sweeping continuously, tasting the water for the chemical trails left by marine worms. When a worm is detected, the snail will slowly approach, sometimes partially burying itself, to reduce its profile before extending its proboscis and delivering a precise, rapid strike. The venom of vermivores is tailored to the physiology of worms, often inducing a rapid, sustained contraction or flaccid paralysis that prevents the worm from burrowing back into the sediment or escaping into crevices. The snail then slowly engulfs the immobilized worm, a process that can be surprisingly protracted as the extensible mouth slowly draws in the entire length of its prey. Mollusk-hunting cone snails or molluscivores face a unique challenge. Their prey, other snails or bivalves, are often protected by their own hard shells or possess strong muscles that allow them to clamp shut. These cone snails have evolved sophisticated ways to overcome these defenses. Some molluscivores might use their siphon to nip at the siphon of another snail, delivering a quick jab of venom to paralyze it before it can fully retract. Others may target the soft tissues exposed when a bivalve or another snail partially emerges from its shell. Still others might use their venom to induce a relaxation of the prey's adductor muscles, allowing them to pry open the shells of bivalves. The conotoxins in molluscivorous venoms are highly specific, designed to disrupt the nervous and muscular systems of other mollusks, demonstrating a remarkable biochemical arms race between predator and prey within the same class of animals. In all these diverse hunting strategies, the underlying principles remain consistent: stealth, precision, and the overwhelming power of the venom. The cone snail's shell provides the initial layer of deception, allowing it to blend seamlessly into its environment. The highly sensitive chemoreceptors of its siphon provide the acute sensory input guiding the hunt. The extensible proboscis and the specialized Radular tooth provide the mechanical delivery system. And the venom that liquid labyrinth of peptides delivers the final decisive blow, ensuring that the prey, once vibrant and alive, succumbs to a silent, paralytic embrace. The efficiency of these hunting strategies is a testament to millions of years of co-evolution, a continuous refinement of both offensive and defensive mechanisms in the marine ecosystem. Each successful hunt reinforces the genetic traits that contribute to the snail's predatory prowess, ensuring that this beautiful, silent killer continues to thrive in its watery domain. The entire process, from the first faint chemical whisper detected by the siphon to the final slow engulfment of the paralyzed prey, is a mesmerizing display of nature's intricate design. A quiet drama unfolding beneath the waves, a silent testament to the cone snail's profound and often deadly purpose. As we allow our thoughts to continue their gentle drift, carrying with us the understanding of these silent hunts, we are now prepared to consider the moments when this ancient, specialized weapon unexpectedly encounters an altogether different and often tragic target: the unwary human. Human vulnerability to the silent sting. As the silent predatory strategies of the cone snail unfold beneath the waves, a profound and often tragic irony sometimes emerges from the sun-drenched beauty of tropical shores. For humans, the encounter with this creature is almost always an unwitting one, born not of malice or aggression, but from a simple, deep-seated human curiosity, a primal fascination with the ocean's shimmering jewels. The shells, so exquisitely patterned and aesthetically pleasing, beckon to the hand, promising a tangible piece of natural artistry, a memento of a tranquil day by the sea. It is in this innocent act of reaching, of touching, of collecting, that the silent hunter's spear can find an altogether different and profoundly vulnerable target. Imagine a scene of serene beauty, a pristine white sand beach stretching lazily beneath the warm embrace of a tropical sun. The water, translucent and inviting, laps gently at the shore, revealing a myriad of wonders beneath its surface. A swimmer, perhaps, or a curious beachcomber wades through the shallow, sun-dappled expanse, their eyes scanning the seabed for treasures. A vibrant coral reef shimmers in the distance, a kaleidoscope of life and color. And then, partially buried in the fine, yielding sand, or perhaps nestled among the smooth, water-worn pebbles, a shell of exceptional grace catches the eye. Its patterns are intricate, a delicate mosaic of earthy browns, creamy whites or subtle violets, promises of a quiet elegance held within its smooth conical form. It appears utterly still, a discarded relic, a silent testament to the ocean's artistry. The hand descends slowly, deliberately drawn by an almost irresistible urge to possess this natural masterpiece. The fingers gently close around the cool, smooth surface, lifting it from its watery bed. There is a moment of pure, unadulterated wonder, a quiet satisfaction in holding such a perfect specimen. And then, a fleeting sensation. A pinprick, perhaps so fine and subtle that it might be dismissed as a tiny fragment of coral, a stray splinter, or the sting of a minuscule jellyfish. It is not the immediate searing pain of a bee sting, nor the sharp, throbbing agony of a stonefish. It is often a quiet, almost dismissible sharpness, a momentary intrusion that might barely register in the immediate aftermath of discovery. There is no visible wound, no dramatic gush of blood, no overt sign of the profound event that has just transpired. The shell remains beautiful and inert in the hand. But beneath that initial deceptive innocuousness, the silent work has already begun. The cone snail, disturbed from its quiet repose, has swiftly extended its proboscis, delivering its hollow harpoon-like Radular tooth, and with it, a precise, potent dose of its specialized venom. The initial sensation, that fleeting pinprick, might soon give way to a subtle numbness, a creeping dullness that begins at the sight of the sting. This numbness, often described as a localized paresthesia, spreads slowly, insidiously through the fingertips, up the palm, and into the wrist, a sensation akin to an extremity falling asleep but without the tingling recovery. It is the insidious work of the conotoxins already beginning to disrupt the delicate balance of nerve function, particularly the voltage-gated sodium channels that govern the transmission of sensory signals. As minutes turn into an hour, or perhaps even less, depending on the species of cone snail, and the amount of venom injected, the symptoms begin to deepen and broaden their embrace. The initial numbness might give way to a dull, aching pain radiating from the sting site, a discomfort that grows steadily, unrelentingly. The area around the wound, though often still outwardly unremarkable, might become swollen tender to the touch, and perhaps slightly discolored. But it is the systemic effects, the far-reaching impact on the body's vital systems, that truly signal the profound danger. A feeling of generalized weakness might begin to creep over the victim, a sudden, inexplicable lassitude that drains the strength from limbs. Dizziness might set in, a disorienting sensation of unsteadiness, as if the ground itself were subtly shifting. Vision might become blurred, the world appearing hazy or indistinct, as the conotoxins begin to affect the intricate neural pathways controlling ocular function. Speech might become slurred, the words difficult to form, the tongue feeling heavy and uncooperative. Swallowing can become arduous, a simple sip of water feeling like an insurmountable task, as the muscles of the throat lose their precise coordination. These progressive symptoms are a direct consequence of the conotoxins diligently finding their molecular targets throughout the human nervous system. The alpha conotoxins, for instance, silently bind to the nicotinic acetylcholine receptors at the neuromuscular junctions, those crucial communication points between nerves and muscles. As these receptors are blocked, the nerve's command to contract the muscles cannot be received. This leads to the profound muscle weakness, the difficulty in moving limbs, the slurring of speech, and most critically, the impairment of the diaphragm and intercostal muscles, which are absolutely essential for breathing. The victim might experience increasing shortness of breath, a desperate struggle for air, as the very muscles that power respiration falter and cease their rhythmic work. This respiratory paralysis is the most common cause of fatality in severe cone snail envenomations. Muconotoxins, those silent blockers of voltage-gated sodium channels, contribute to the widespread numbness and weakness, preventing nerve impulses from traveling effectively. Omega-conotoxins disrupt the release of neurotransmitters, further compounding the communication breakdown across the nervous system, leading to a profound, widespread neurological dysfunction. The synergy of these diverse peptides, each acting on a different vital pathway, overwhelms the body's defenses, leading to a rapid and cascading failure of neurological and muscular control. Human vulnerability to the silent sting is magnified by several crucial factors. Foremost among them is a profound lack of awareness. Many people, even seasoned beachcombers or experienced divers, are simply unaware of the deadly potential hidden within these beautiful shells. They see only the aesthetic appeal, the quiet grace, and not the formidable predator within. The harmless appearance of a shell, often inert and seemingly lifeless, belies the active venomous creature that can emerge with astonishing speed when disturbed. Furthermore, the potency of the venom varies significantly, between different cone snail species. Some, like the Conus geographus, are notoriously lethal, responsible for the majority of recorded human fatalities due to their highly potent, fast-acting neurotoxic venom cocktail specifically evolved to paralyze fish almost instantly. Others, such as Conus textile or Conus marmorius, also possess venoms capable of causing severe envenomation, though perhaps with a slightly slower onset or a lower fatality rate depending on the dose injected. The amount of venom injected can also vary. A partial strike might deliver a lesser dose, while a full sustained injection can be devastating. Individual physiological responses also play a critical role. Factors such as the victim's age, overall health, and body mass can influence the severity of the reaction. Children, being smaller, are often more susceptible to a given dose of venom, as are individuals with pre-existing heart or respiratory conditions. The immediate availability of medical aid is another critical determinant of outcome. Many encounters occur in remote tropical locations far from hospitals, equipped to handle such specialized toxicological emergencies, where access to artificial respiration or other life-sustaining measures might be delayed or unavailable. Tragic reports from tropical shores spanning decades underscore this vulnerability. Tales of shell collectors, divers, or even children playing in shallow waters, succumbing to the silent sting, serve as somber reminders of the profound power concealed within these oceanic jewels. The victims often report a progressive decline from a mild localized sensation to profound systemic paralysis, culminating in respiratory failure, if medical intervention is not swift and effective. There is no anti-venom readily available for cone snail stings, meaning treatment is largely supportive, focusing on maintaining breathing and other vital functions until the body can metabolize and clear the toxins. The silence of the threat is perhaps its most chilling aspect. Unlike a snake that might hiss or a jellyfish that delivers an immediate burning pain, the cone snail offers no overt warning. Its beauty is its most effective lure, its initial sting a mere whisper. It is a stark reminder that in the intricate ballet of marine life, the most captivating forms can conceal the most profound dangers, demanding respect and a quiet vigilance from those who venture into their ancient watery domain. As we allow our thoughts to drift, carrying with us the weight of these unwitting encounters, we are now prepared to explore a remarkable paradox: how the very venom that brings such peril can also hold the promise of profound healing, transforming from lethal to life-saving in the hands of scientific inquiry. Conotoxins in modern medicine. Where the silent spear once brought stillness and its liquid essence whispered promises of profound, often fatal paralysis to the unsuspecting, an astonishing transformation now takes place in the quiet laboratories of human ingenuity. The very conotoxins, those complex peptides responsible for the cone snail's deadly embrace, are being meticulously studied, purified, and reimagined not as agents of harm, but as exquisitely precise tools for healing. This profound paradox, the journey from lethal to life-saving, represents one of the most compelling frontiers in modern medicine, where the venom of a seemingly simple marine mollusk holds the potential to alleviate human suffering in ways that traditional pharmaceuticals often cannot. The key to this remarkable transformation lies in the unparalleled specificity of conotoxins. Unlike many conventional drugs that might interact broadly with various targets in the body, leading to a cascade of undesirable side effects, each conotoxin peptide is a molecular key, crafted by millions of years of evolution to fit a particular lock, a specific ion channel, or a precise neurotransmitter receptor within the nervous system. This exquisite selectivity, which allows the cone snail to incapacitate its diverse prey with such efficiency, is precisely what makes these peptides so invaluable to medical science. They offer a level of precision targeting that is often unmatched by synthetic compounds, allowing researchers to modulate specific neural pathways with minimal collateral damage. One of the most significant breakthroughs in this field has been the development of ziconotide, a synthetic version of an omega conotoxin originally isolated from the venom of Conus magus, the magic cone snail. This remarkable compound approved for human use is a potent non-opioid pain reliever specifically prescribed for severe chronic pain that is refractory to other treatments, particularly neuropathic pain, which arises from damage to the nervous system itself. Imagine the relentless burning agony of nerve pain, a torment that can persist for years, often unresponsive to even the strongest opioid medications, which themselves carry the heavy burden of addiction and debilitating side effects. Ziconotide offers a beacon of hope in such desperate circumstances. Its mechanism of action is a testament to the conotoxin's precision. Ziconotide selectively blocks N-type voltage-gated calcium channels. These particular calcium channels are found predominantly on the terminals of primary afferent neurons, the nerve cells that transmit pain signals from the periphery of the body up to the spinal cord and then to the brain. When a pain signal arrives at these nerve terminals, N-type calcium channels open, allowing calcium ions to rush in, which in turn triggers the release of neurotransmitters, the chemical messengers that relay the pain signal to the next neuron in the pathway. By blocking these specific channels, ziconotide effectively prevents the release of these pain-signaling neurotransmitters. It is like placing a tiny invisible plug into the very conduits that allow pain messages to be sent, thereby silencing the signal before it can reach the brain's perception centers. The beauty of this action is its localized effect. Ziconotide is typically administered directly into the spinal fluid via an intrathecal pump, allowing it to act precisely where pain signals are processed in the spinal cord, minimizing systemic exposure and greatly reducing the risk of the widespread side effects associated with orally administered pain medications. This targeted delivery, combined with its non-opioid nature, means it does not cause respiratory depression, sedation, or addiction, which are common and serious concerns with traditional opioid analgesics. Beyond pain management, the vast, unexplored diversity of conotoxins holds immense promise for a wide array of neurological disorders. Scientists are currently investigating other omega conotoxins for their potential in treating epilepsy, a condition characterized by abnormal synchronized electrical activity in the brain. By subtly modulating various calcium channels, these peptides might help to stabilize neuronal excitability, preventing the uncontrolled electrical storm that defines an epileptic seizure. Imagine a future where a precisely engineered conotoxin could gently dampen the overactive circuits in the brain, restoring a quiet, rhythmic balance without the broad suppressive effects of current anti-epileptic drugs. Alpha conotoxins, those peptides that so effectively target nicotinic acetylcholine receptors, are also generating significant excitement in the realm of neurodegenerative diseases. These receptors play complex roles in memory learning and cognitive function. Researchers are exploring whether certain alpha conotoxins, perhaps modified or used in very low controlled doses, could selectively enhance or protect specific subtypes of nicotinic receptors, potentially offering new therapeutic avenues for conditions like Alzheimer's disease or Parkinson's disease, where cognitive decline and motor dysfunction are prominent features. The hope is that these peptides could help to restore lost neural communication or protect vulnerable neurons from damage, slowing the relentless progression of these debilitating illnesses. the liquid labyrinth extends further into the realm of stroke and traumatic brain injury. Some conotoxins are being studied for their potential neuroprotective properties. Following a stroke, a cascade of damaging events, including excessive release of excitatory neurotransmitters like glutamate, can lead to widespread neuronal death. Certain conotoxins, by subtly modulating specific ion channels or receptors, might be able to dampen this excitotoxic cascade, protecting brain cells from secondary damage and potentially improving recovery outcomes. This involves understanding how these peptides could stabilize neuronal membranes or reduce the overstimulation that leads to cell death, offering a quiet shield against the devastating aftermath of neurological trauma. The potential applications are not confined to the nervous system alone. Research is also delving into the possibility of using conotoxins as novel agents in cancer therapy. Certain ion channels, particularly voltage-gated sodium channels and calcium channels, are known to be overexpressed or aberrantly active in various cancer cells, playing roles in their proliferation, migration, and metastasis. By selectively targeting these specific channels in cancer cells, conotoxins could potentially inhibit tumor growth, prevent the spread of cancer to other parts of the body, or even make cancer cells more susceptible to conventional chemotherapies. Imagine a molecular sniper, a conotoxin engineered to seek out and disrupt the vital channels that fuel cancer's relentless spread, leaving healthy cells largely untouched. Beyond their direct therapeutic applications, conotoxins are also invaluable tools for fundamental scientific research. Their unparalleled specificity for different ion channel and receptor subtypes makes them ideal molecular probes. By using purified conotoxins, scientists can precisely label, identify and study the distribution and function of specific channels and receptors in various tissues, providing critical insights into the intricate workings of the nervous system and other physiological processes. This fundamental understanding is essential for designing new drugs and therapies, allowing researchers to map the complex circuitry of life with unprecedented detail. The journey from a deadly venom to a life-saving medicine is, however, a long and arduous one fraught with scientific challenges. The intricate chemical synthesis of these complex peptides, ensuring their structural integrity and biological activity, requires advanced biochemical techniques. Delivering these peptides effectively to their target sites within the body, especially across the blood-brain barrier for central nervous system disorders, presents significant hurdles. Furthermore, meticulous clinical trials are required to establish their safety, efficacy, and optimal dosing in human patients. The potential for unintended side effects, even with highly specific molecules, must be rigorously evaluated. Yet the promise remains immense. The ocean, that vast, ancient reservoir of life, continues to reveal its secrets, and among its most astonishing offerings are the conotoxins, those molecular keys crafted by the cone snail. They represent a treasure trove of biochemical diversity, a liquid labyrinth whose pathways, once understood, can lead not to paralysis, but to profound relief and healing. As scientists continue to unravel the intricate mechanisms of these peptides, translating their silent, deadly power into precise therapeutic interventions. The cone snail stands as a quiet testament to nature's boundless capacity for both creation and destruction. A creature whose venom, once feared, now whispers promises of a future where suffering might be gently eased one molecular key at a time. And as our thoughts continue their gentle drift, carrying with us the understanding of this remarkable transformation, we are now prepared to explore the broader ecological role of these silent architects within their vibrant, watery homes. The cone snail's ecological role. As our gentle journey continues to unfold, drifting past the astonishing intricacies of venom and the profound medical promise it holds, we now turn our attention to the wider stage upon which the cone snail performs its silent ancient drama, the vibrant, bustling ecosystem of the coral reef. These underwater cities teeming with life in a kaleidoscope of forms and colors are not merely collections of individual creatures, they are intricate, interconnected webs, each thread of life playing a crucial role in maintaining the delicate, pulsing balance of the whole. Within this complex dynamic system, the cone snail, with its deceptive beauty and potent venom, emerges not merely as a solitary hunter, but as a quiet, indispensable guardian, a vital cog in the grand machinery of the reef. Coral reefs, those magnificent living structures, are often described as the rainforests of the sea, harboring an extraordinary diversity of species. Yet, like any complex community, they require constant regulation to prevent any single population from overwhelming the others. This is where predators like the cone snail step into their essential ecological role. By preying upon fish, marine worms, and other mollusks, cone snails act as natural population control agents, ensuring that no single species grows unchecked, thereby preventing resource depletion and maintaining the overall health and stability of the reef community. Imagine a delicate scale constantly being balanced by the subtle shifts of life and death, and the cone snail is one of the unseen hands subtly adjusting the weights. The sheer diversity of cone snail species, estimated at around 800 distinct forms, is itself a testament to their specialized ecological niches. Each species, often with its uniquely tailored venom cocktail and specific hunting strategy, tends to specialize in a particular type of prey. Some cone snails are dedicated piscivorous, focusing almost exclusively on fish, while others are vermivores expertly hunting marine worms, and still others are melusivores preying on other snails or bivalves. This remarkable specialization means that different cone snail species are exerting predatory pressure on distinct segments of the food web. This prevents a single predator from decimating one particular prey population, while simultaneously ensuring that a broad spectrum of prey species are kept in check. It is a subtle, distributed form of ecological regulation, ensuring that the reef's resources are utilized efficiently and that competitive pressures remain balanced. Consider the swift, darting movements of small reef fish. Without predators like the piscivorous cone snails, certain species of fish might proliferate excessively, potentially overgrazing algal beds or outcompeting other fish for limited food resources, thereby altering the delicate structure of the reef. The sudden paralytic strike of a cone snail serves as a powerful, albeit localized check on these populations. Similarly, the vast and often unseen communities of marine worms, burrowing through the sand and sediment, play crucial roles in nutrient cycling. However, an overabundance of certain worm species could disrupt these processes. The vermivorous cone snails, with their precise worm-targeting venoms, help to regulate these populations, ensuring that their ecological contributions remain within sustainable bounds. Even other mollusks which might otherwise compete with the cone, snails themselves for space or food, are kept in check by the molluscivorous species, creating a dynamic equilibrium where no single group dominates entirely. Beyond simple population control, the predatory actions of cone snails also contribute to the overall fitness and resilience of prey populations. Predators, by their very nature, often target the weaker, slower, or less vigilant individuals within a prey group. This natural selection pressure ensures that only the fittest, most adaptable individuals survive to reproduce, passing on their advantageous traits to subsequent generations. In this quiet, ancient struggle, The cone snail inadvertently acts as an evolutionary sculptor, continuously refining the genetic makeup of its prey, making the entire ecosystem more robust and capable of adapting to environmental changes. It is a harsh, yet ultimately beneficial process that ensures the long-term vitality of the reef. While primarily recognized as predators, cone snails are also, albeit less frequently, part of the prey spectrum themselves. Their robust, often heavily calcified shells provide a formidable defense against many potential attackers. However, larger, more powerful predators such as certain species of crabs with crushing claws, octopuses with their intelligent manipulation, or some specialized fish with powerful jaws are known to occasionally prey on cone snails. The snail's ability to quickly retract its entire soft body deep within its shell, relying on the strength of its outer dwelling, is its primary defense against such threats. This interaction, though less common, means that cone snails are also integrated into the food web from both sides, contributing to the energy flow and nutrient cycling within the reef ecosystem, ensuring that the energy locked within their bodies is eventually returned to the broader biological community. Furthermore, the very process of shell formation by cone snails, the slow, continuous deposition of calcium carbonate, plays a subtle but significant role in the biogeochemical cycles of the reef. These shells, when the snail eventually perishes, contribute to the vast accumulation of biogenic carbonates that form the very fabric of coral reefs and sandy seabeds. Over geological timescales, these shell fragments become part of the sediment, eventually compacting and contributing to the formation of limestone, thereby playing a role in the long-term sequestration of carbon from the ocean, a silent contribution to the planet's grand elemental cycles. The presence and diversity of cone snail populations are also often considered by marine biologists as indicators of reef health. A thriving, diverse population of cone snails suggests a healthy, balanced ecosystem with ample prey resources and stable environmental conditions. Conversely, a decline in cone snail numbers or species diversity might signal underlying environmental stresses such as pollution, habitat, degradation, or changes in water quality, which could be impacting the broader reef community. They are, in a sense, quiet sentinels, their well-being reflecting the vitality of their complex homes. Yet these silent guardians are not immune to the pervasive reach of human activities. Overcollection of their beautiful shells, driven by aesthetic demand, can locally deplete populations, disrupting the delicate balance they maintain. Pollution, particularly chemical runoff from land-based activities, can directly impact their sensitive chemosensory abilities, impairing their hunting success or even directly poisoning them. Climate change, with its accompanying ocean acidification and rising sea temperatures, poses a profound threat to the entire coral reef ecosystem, and by extension, to the cone snails that depend on these habitats. Ocean acidification in particular makes it more difficult for mollusks to form and maintain their calcium carbonate shells, potentially weakening their primary defense and increasing their vulnerability. Thus, the cone snail, often perceived solely through the lens of its individual beauty or its potential danger, is in fact a crucial interconnected component of the marine world. Its predatory actions, driven by the astonishing power of its venom, are not merely acts of survival for the individual, they are vital contributions to the ecological stability and biodiversity of the coral reef. These creatures, with their quiet patience and their precise chemical warfare, help to sculpt the very fabric of life beneath the waves, ensuring that the intricate dance of predator and prey continues in a delicate, ancient rhythm. They are indeed guardians of the reef, not through active protection, but through the profound and often unseen consequences of their existence, maintaining the subtle equilibrium that allows these vibrant underwater cities to flourish. As we allow our thoughts to continue their gentle drift, carrying with us the image of these silent patterned guardians, we are now prepared to delve into the even deeper currents of time to explore the evolutionary masterpiece that crafted such specialized weapons and such deceptive beauty. The genesis of a specialized weapon. In the boundless ocean of geological time where aeons unfold like the slow rhythmic breath of the deep, the story of the cone snail's specialized weapon began to etch itself into the very fabric of life. To truly appreciate this evolutionary masterpiece, we must journey back hundreds of millions of years to the ancient origins of gastropods, a vast and diverse class of mollusks that includes everything from garden snails to sea slugs. The earliest gastropods were humble creatures often equipped with a simple, ribbon-like radula, adorned with numerous rows of small, chitinous teeth. This ancestral radula served as a versatile scraping tool, perfectly suited for grazing algae from rocks or rasping detritus from the seabed, a gentle, almost passive existence. But within this ancient lineage, a profound divergence began, a quiet revolution, driven by the relentless pressures of survival and the endless possibilities of adaptation. Scientists theorize that as certain gastropod ancestors ventured into more competitive environments, perhaps where food resources were scarce or where active prey was abundant, the benefits of a more predatory lifestyle began to emerge. It was here in this crucible of natural selection that the radula, that unassuming scraping ribbon, started its astonishing transformation. Imagine the subtle incremental steps over countless generations. Perhaps certain teeth on the radula in some ancestral species became slightly larger, a little sharper, allowing for a more effective grasp, or a rudimentary, piercing action. Then, as mutations accumulated, these specialized teeth might have begun to detach from the main radula ribbon, becoming individual disposable units. This detachment was a critical innovation, freeing the tooth to be used as a projectile rather than a fixed rasp. Concurrently, the internal musculature surrounding these teeth would have evolved, gaining the capacity for precise, forceful deployment. The hollowing of the radula tooth was another pivotal leap. Initially, a solid tooth might have inflicted a wound, but a hollow tooth opened the door to a far more potent strategy: the injection of incapacitating substances. This required the co-option and specialization of an existing gland. Most gastropods possess salivary glands which typically produce digestive enzymes or lubricating secretions. Scientists believe that in the evolving cone, snail lineage, one of these ancestral salivary glands gradually began to produce compounds that were not merely digestive, but mildly toxic, perhaps initially, to subdue struggling prey or to aid in external digestion. Over millions of years, through a process of gene duplication and diversification, this gland would have transformed into the highly specialized venom gland. We observe today a miniature biochemical factory dedicated to synthesizing a complex array of neurotoxins. Gene duplication is a fundamental engine of evolution, particularly in the creation of complex venom systems. Imagine a gene responsible for producing a particular protein or peptide. Through an error in DNA replication, an entire copy of this gene might be created. Now, the organism has two copies. One copy can continue to perform its original vital function, ensuring the creature's survival. The other copy, however, is now free to accumulate mutations without immediately compromising the organism. Most of these mutations would be detrimental or neutral, but occasionally a mutation would arise that grants the duplicated gene a new, advantageous function, perhaps producing a peptide with a slightly different shape, capable of interacting with a different receptor in the prey or binding more tightly to an existing one. This process, repeated countless times over vast stretches of geological time, led to the astonishing diversity of conotoxins, where a single cone snail species can produce hundreds or even thousands of distinct peptides, each a finely tuned molecular key. This diversification of venom was inextricably linked to the escalating evolutionary arms race between predator and prey. As cone snails developed more potent toxins, their prey, fishworms and other mollusks, would have simultaneously evolved counter-defenses, perhaps developing resistance to certain toxins or adapting behaviors to evade detection. This continuous pressure from both sides drove an ever-increasing specialization in the cone snail's venom, leading to the highly specific conotoxins that target particular ion channels and receptors unique to their chosen prey. A toxin that effectively paralyzes a fish might be entirely ineffective against a marine worm, and vice versa. This is why different cone snail species have evolved distinct venom cocktails and unique Radular tooth morphologies, each precisely adapted to overcome the specific physiological defenses of their preferred diet. The adaptive radiation of cone snails, resulting in the approximately 800 known species today, is a testament to this profound evolutionary journey. From a common ancestor, different lineages diversified, each specializing in a particular feeding strategy. We see the emergence of piscivores with their lightning-fast, highly potent venoms designed for rapid fish paralysis, and robust barbed harpoon teeth to secure slippery, struggling prey. Then there are the vermivores, whose venoms are tailored to immobilize marine worms and whose slender, often serrated teeth are perfectly suited for piercing soft, segmented bodies. And the molluscivores, facing the unique challenge of other armored mollusks, evolved venoms to overcome their specific defenses, perhaps inducing muscle relaxation or targeting unique neural pathways in their shelled relatives. Each of these specialized diets drove further refinement in venom composition, tooth shape, and hunting behavior, carving out distinct ecological niches within the reef environment. The evolution of the cone snail's sophisticated sensory system also played a crucial role in this masterpiece. The development of the elongated, highly chemosensitive siphon, capable of tasting the water for faint chemical plumes of prey from a distance, was a critical adaptation for a slow-moving ambush predator. This allowed the snail to detect its prey without relying on sight, which is often limited in the murky, complex underwater environment, or without the need for active pursuit, which would expend too much energy. The ability to sense the subtle molecular whispers of a fish or a worm long before it comes into visual range gave the cone snail a profound advantage, allowing it to prepare its silent ambush with perfect timing. Even the shell, that beautiful, deceptive outer dwelling, underwent its own evolutionary sculpting. Its conical form, while providing robust protection, also evolved intricate patterns and colors that are not merely aesthetic. These patterns are a masterclass in camouflage, meticulously designed to help the snail blend seamlessly into the dappled light, and complex textures of coral reefs, sandy seabeds, and rubble zones. This visual deception is an integral part of the hunting strategy, allowing the snail to remain hidden, an almost invisible presence, until the precise moment of strike. The shell's evolution, therefore, was intertwined with the development of the venom apparatus, creating a synergistic system where beauty, stealth, and lethal power converge. The genesis of this specialized weapon, the cone snail's venom system, is a profound narrative of evolutionary innovation. It speaks to the incredible capacity of life to adapt to repurpose existing structures and genes, and to refine biological mechanisms over unimaginable stretches of time. From a simple scraping gastropod through a relentless process of natural selection, gene duplication, and targeted mutation, emerged a creature of astonishing complexity and predatory prowess. It is a living testament to the power of an evolutionary arms race, where the constant pressure from prey drove the development of ever more sophisticated and specific tools of chemical warfare. The cone snail, in its quiet existence, embodies millions of years of biological refinement, a masterpiece sculpted by the currents of time, its specialized weapon, a silent, enduring marvel. As we allow our thoughts to continue their gentle drift, carrying with us the deep understanding of this creature's ancient past, we are now prepared to gaze into the future, to ponder the unanswered questions and the potential discoveries that still lie hidden within the depths of its remarkable biology. Unanswered questions and future discoveries. As our journey through the intricate world of the cone snail gently approaches its quiet horizon, we carry with us a profound understanding of its deceptive beauty, its specialized anatomy, its astonishing venom, and its vital ecological role. Yet, in the boundless expanse of scientific inquiry, every answer often illuminates a constellation of new questions, and the cone snail, in its ancient wisdom, still holds many secrets close to its silent, patterned heart. The whispers from the abyss continue beckoning us to explore the vast, unexplored territories of its biology and the untold stories yet to be discovered within its liquid labyrinth. One of the most profound mysteries still unfolding is the sheer unquantified diversity of conotoxins themselves. While thousands of distinct peptides have already been identified and characterized, scientists believe that this represents merely a fraction of the full biochemical repertoire. Imagine the vast, largely unexplored depths of the tropical oceans, the countless coral reefs, rocky shores, and sandy expanses that remain untouched by human hands, each potentially harboring unique cone snail species, each with its own finely tuned, species-specific venom cocktail. Each new species discovered, each new population sampled, opens another molecular door, revealing a novel set of peptides, each a unique key designed to unlock a specific biological lock. The quest to fully catalogue this astonishing biochemical diversity is a monumental undertaking, a continuous patient sifting through the ocean's genetic library, searching for the next transformative compound. Beyond simply identifying new conotoxins, a deeper mystery lies in understanding the precise mechanisms of their synergy. We know that the cone snail's venom is not a single toxin, but a complex orchestra of hundreds, sometimes thousands of different peptides, each playing its own subtle part. But how do these individual notes combine to create such a devastating symphony of paralysis? How do they interact with each other within the prey's body, amplifying each other's effects, or perhaps even modulating the action of other toxins in ways we do not yet fully comprehend? Is there a precise sequence of molecular attacks, a choreographed dance of peptides that leads to such rapid and overwhelming incapacitation? Unraveling these intricate synergistic relationships is like deciphering a complex musical score, understanding not just the individual instruments but the harmonious and dissonant interplay that creates the profound overall effect. This understanding could unlock new strategies for drug design, where combinations of compounds rather than single molecules might offer more potent and targeted therapeutic outcomes. Another fascinating yet largely unanswered question revolves around the dynamic regulation of venom production within the snail's own body. We know that the venom gland acts as a miniature pharmaceutical factory, but how does the snail precisely control its output? Do environmental cues such as temperature fluctuations or changes in prey availability trigger alterations in the venom's composition? Can a cone snail over its lifetime subtly adjust the ratio or even the types of conotoxins it produces, adapting its biochemical arsenal to the specific challenges of its immediate environment or the changing dietary needs of its developmental stages. Scientists theorize that such dynamic control would represent an astonishing level of biological sophistication, allowing the snail to fine-tune its weapon with remarkable adaptability, a living, evolving chemical laboratory constantly optimizing its output for maximum predatory success. Uncovering these regulatory mechanisms could offer insights into how biological systems adapt their chemical defenses with such precision. The deep evolutionary pathways that sculpted this specialized weapon also continue to hold many secrets. Tracing the exact genetic changes, the precise gene duplications, and the subtle mutations that transformed an ancestral scraping radula into a hollow venom-injecting harpoon and a simple salivary gland into a potent venom factory is an ongoing endeavor. Paleontological records, though sparse for soft-bodied creatures combined with advanced genomic analyses, are slowly beginning to piece together this ancient narrative. By comparing the genomes of various cone snail species and even related gastropods, researchers can reconstruct the molecular history of this evolutionary masterpiece, identifying the key genetic innovations that led to the genesis of such a formidable and diverse chemical arsenal. This deep dive into the past illuminates not only the cone snail's story, but also the broader principles of evolutionary innovation itself. Beyond the venom, many aspects of the cone snail's fundamental biology and ecology still remain cloaked in mystery. What are the subtle behavioral nuances of less studied species, particularly those inhabiting deeper, less accessible marine environments? How do these creatures, often moving with such deliberate slowness, navigate the vastness of the ocean floor, locate their mates, or avoid their own predators? The intricate details of their reproductive cycles, particularly the pelagic larval stages of many species, are still largely unknown. How do these tiny drifting larvae survive the vast planktonic journey, disperse across wide geographical ranges, and eventually settle into their specific adult habitats? These early life stages are critical bottlenecks for population survival, and understanding them is vital for conservation efforts. And what of the cone snails that reside in the true abyss, in the crushing pressures and perpetual darkness of the deep sea? Are there species lurking in these extreme environments with venoms even more potent, or perhaps with entirely novel biochemical properties adapted to incapacitate creatures of the deep? The exploration of these abyssal zones represents a frontier of discovery, promising to reveal new forms of life and new biochemical wonders, potentially including cone snails whose venoms could hold even greater therapeutic promise. Looking towards the future, the potential discoveries emanating from cone snail research are truly boundless. Beyond the established pain relief of zyconotide, scientists are actively exploring other conotoxins for their potential to treat an ever-expanding list of human ailments. Imagine a future where conotoxins could be precisely engineered to target specific cancer cells, inhibiting their growth or preventing their spread without harming healthy tissues. What about their potential in autoimmune diseases, subtly modulating immune responses that have gone awry? Or in treating infectious diseases by disrupting the vital processes of pathogens? Could they serve as novel diagnostic tools, molecular probes that can precisely identify biomarkers for disease in the earliest stages? The possibilities stretch as far as the imagination allows, fueled by the exquisite specificity and diversity of these natural compounds. The advent of omics technologies, genomics, proteomics, transcriptomics, is revolutionizing our ability to unravel the cone snail's secrets. These technologies allow researchers to rapidly sequence entire genomes, identify every protein produced, and map the gene expression patterns within the venom gland. This comprehensive molecular understanding will undoubtedly accelerate the discovery of new conotoxins, elucidate their evolutionary origins, and reveal the intricate regulatory networks that govern their production. Furthermore, the very architecture of the Radular tooth, that hollow barbed spear, could inspire new designs in bio-inspired engineering, leading to the development of advanced microneedles for targeted drug delivery or novel surgical instruments that mimic nature's precision. Finally, the future of cone snail research is inextricably linked to the urgent need for conservation. As we uncover more of their profound secrets, we also become more aware of the threats facing these creatures and their fragile coral reef habitats. Understanding their life cycles, their ecological roles, and their genetic diversity is paramount to developing effective conservation strategies, ensuring that these remarkable architects of the deep continue to thrive, not only for their intrinsic value, but also for the immeasurable potential they hold for future scientific and medical breakthroughs. The whispers from the abyss continue a gentle invitation to listen, to learn, and to protect the silent wonders that still unfold beneath the waves. And so, as we drift away from the intricate world of the cone snail, we carry with us the quiet understanding of how profound beauty can conceal astonishing power, much like the hidden depths of our own minds. Just as the ocean's gentle currents carry its patterned jewels to silent repose, so too may your thoughts, now softened and diffused, be carried into the tranquil embrace of slumber. Let the intricate, unseen patterns of the deep guide your consciousness into a state of profound rest, where the mysteries of the subconscious begin to unfold their own quiet narratives. May the gentle, rhythmic hush of the ocean cradle your spirit, and may your dreams be as serene and intricate as the most beautiful silent shell holding its own profound secrets. Allow your mind to sink slowly deeply into the quiet, reassuring darkness, letting all conscious thought dissolve like sand into the soft, yielding seabed as you are drawn deeper into the liquid labyrinth of your own peaceful dreams. May the silent currents of the deep carry you to a truly restorative rest. Sleep well, goodbye, and see you again.